Good afternoon, everyone. Thank you for the opportunity for me to introduce Coeptis to you all. I am the Chief Scientific and Chief Medical Officer there. This is the obligatory slide that you can read really quickly. Okay, so Coeptis Therapeutics, sorry, I need to be able to see this. Coeptis Therapeutics is a clinical stage biotechnology company located in Pittsburgh, Pennsylvania. However, we also now have outreach into the Seattle area with PD and in-house PD and R&D efforts there. So like all of you sitting here today and the talks that you've all been hearing, I think we share a common goal in bringing cell and gene therapy to our patients and accelerating these therapies. And what I'm going to talk to you today about is really the approach that we are taking in really trying to bring together some first-in-class innovative technologies to really address, hopefully, some of the hurdles that people are facing in the allogeneic cell therapy space. So in the time allotted, it's obviously not very much time, so I'm going to give you a very high-level overview of the key assets that we have, including a recent licensed proprietary cell expansion and differentiation platform from Devera Therapeutics, where I developed this technology. Also, an unmodified NK cell therapeutic called DVX-201, so I'll talk about these two things. And then finally, some engineering, really interesting and novel engineering approaches. One is a SNAP CAR approach. This is a universal CAR technology. And then finally, I'll talk to you about our GEAR NK editing approaches. So the SNAP CAR program is licensed from the University of Pittsburgh out of Jason Lohmuller's lab, and then the Karolinska Institute, we're working with them on development of this GEAR NK approach, and this was licensed out of Vicelix. Okay, so the recent transaction that brought our two companies together is shown on this slide. So this is just an overview of what we are doing here. So we are bringing together very innovative allogeneic cell therapy platforms with an engineering platform to accelerate clinical development. So out of Devera Therapeutics, we have an allogeneic platform that has really been quite well established and used for many years now, with demonstrated clinical safety data and proven ability to expand these CD34 positive cells prior to directing their differentiation. So I'll talk a little bit more about that, and that is being combined with some innovative engineering technologies out of Coeptis, and that's the SNAP CAR and the CD38 GEAR technologies. And really the beauty of bringing these two companies together was the ability to take some of these novel approaches for engineering and put them into an allogeneic proven cell therapy platform to generate next generation SNAP CAR, GEAR NK approaches that are truly universal. Okay, so this transaction also really helped to diversify the pipeline that we see here right now. So this is showing you currently what's in the Coeptis pipeline and the development opportunities that we have. So you can see that we have two phase 1 trials using the unmodified, this is an allogeneic pooled donor approach. This is DVX-201 in relapse refractory AML, as well as high-risk MDS. And then we also did this in the setting of acute viral respiratory diseases. And that was COVID, but clearly that was just proof of concept initially to show that we could do this approach safely. And so I'm not going to talk too much about that one today, but the proof of concept is there for future use in respiratory viruses. And then we also have the CD38 GEAR NK cells for multiple myeloma. And we're looking at both approaches, autologous and allogeneic, in this setting with the Karolinska Institute. SNAP CAR T in the auto setting, SNAP CAR NK in the allogeneic setting. These will be focused on hematologic malignancies as well as solid tumors. And then we have a nice preclinical program in engineering macrophages with proof of concept data. Okay, so before I really launch into some of the bringing together of these two technologies, I do want to talk a little bit about the barriers that we all know to exist in allogeneic cell therapy today. I think these are all well known, and I don't think they're talked about enough, and so I wanted to really bring to the forefront what we are doing to try to overcome these barriers. And these barriers really are related to sourcing of material. We just heard about iPSCs, and we've been hearing a lot about iPSCs. So we can really get source material from iPSC or cord blood or adult-derived material, and I'll talk a little bit more about that. But we also know that the barriers to these living drugs are the donor-to-donor variability that we see, scalability of these processes, Cost of goods, I absolutely agree with what was just said. We have to make these products affordable, and I think the allogeneic approach does do just that. We have to be able to cryopreserve. Not everyone has been able to do this successfully, but this is clearly a critical piece of being able to deliver these products for immediate demand. And then persistence, I think everyone would agree. We know that with allogeneic cell therapies, persistence is a huge problem. And we've really taken this approach to understand that up front and to recognize that we may not have long-term persistence, but we don't necessarily need long-term persistence in order to have efficacy. And I guess I should have said in the beginning, I'm a stem cell transplant physician myself, and all of these things I'm talking about are incredibly important to me as I look at patients and try to consent them for these kinds of procedures. So as we talked about developing these therapies, these are all the things I keep in mind when we do this. Okay, so when we think about these barriers, I'm making a plug for umbilical cord blood here. We have a source of cells that's a beautiful, in my opinion, source of cells. These are sustainable cord blood units. Obviously, babies are born every day. We have FDA licensed banks in the United States, and there are over 800,000 units that have been stored and are regulated. So these are cost-effective, young, healthy sources of hematopoietic stem and progenitor cells. Cord blood is the only source of material that is already FDA regulated. It is a drug under the CFR Section 351. And we know that the storage of cord blood units is validated, and these cells can be banked and held in liquid nitrogen for over 20 years. In fact, I think that number went up to 25 or so years recently, and we know this because these older units have been used in the setting of stem cell transplant, and they do give long-term persistence. So the other things, you're like, okay, well, that's the source material. We obviously use cord blood and are biased towards that source. What about all those other things that I talked about? And one of the ways that we are the way we are overcoming these other areas, these barriers for allogeneic cell therapies is really by using pooled donor manufacturing. So we are the only company that uses a pooled donor manufacturing approach. In fact, we have 3 INDs that have already been through the FDA, and we have been cleared for this type of manufacturing, and we have used these cells in vivo in clinical trials. So we believe that this pooled donor manufacturing program and platform significantly reduces the donor-to-donor variability, the logistics, and the cost. So this provides us with ultimate scalability, right? We can go from 4 units to 20 units and really scale this way. So this improves our yield and it reduces our cost of goods. It provides a consistent and very reliable source of manufacturing to provide true off-the-shelf, and when I say off-the-shelf, I mean non-HLA matched cryopreserved treatments. But again, remember, this is unlike the previous speaker. These are products that we do not expect to persist long-term. And the other thing that I think that bears some discussion is that this approach provides a non-clonal source of products. And this is very important because we would all agree, I think, that allogeneic cell therapies will require multi-dosing approaches. And if you only have a clonal population of cells to give, then you can't give very many doses before that patient will reject it. So by doing pooled donor manufacturing, we hope to overcome that. Okay, so this technology, we're really leveraging the established technology developed at Devera Therapeutics, actually really at the hutch prior to that, and I'm showing you a very high-level overview of that here. So the starting material is pooled donor qualified cord blood units. We do not require HLA matching. We really look at the content of CD34s. We then isolate these CD34 positive cells, and again, this allows for easy scale and eliminates a lot of the donor-to-donor variability. We then expand these cells. This is a very standardized approach that we have utilized, and the expansion of these cells allows us to do progenitor priming prior to differentiation or do early gene editing. So we then go on to differentiate. So this is a directed differentiation phase. In this case, I'm showing you to NK cells. We do a fill and finish. This is a cryopreserved. We know we can cryopreserve and maintain potency and viability. It's a very small dose. It's 20 cc's and can be delivered easily on demand at the bedside. This is the platform for NK cell generation, again, very high level, where we do stem and progenitor cell expansion and priming. So we've optimized all of this. This process has now been through the FDA. There are 2 INDs around this process. We then differentiate these cells. We do not require feeder cells, and that really is because of how we expand the stem and progenitor cells first. We generate our own feeder cell layers for this. And what I'm showing you in the bottom left there is a serial killing assay showing you that we indeed, with one set of NK cells, we can challenge them repeatedly over a number of days and still see repeated killing. And then on the right there, you can see, again, importantly, that we can cryopreserve and maintain viability and cell recovery. What I'm not showing you are the number of in vivo assays that were done. So we have a number of AML tumor models that we've shown excellent results in. So we have two first-in-human, and again, I think this is important. We never have before, so lots of people are doing allogeneic NK cell therapies, but this is the first-in-human of pooled donor unmodified NK cells. And the first trial done was in COVID-19. This was with Dr. Josh Hill at the Fred Hutch Cancer Center and the University of Washington. We did 9 patients, and this was obviously with no lymphodepleting chemotherapy because these patients had viral infections. And it was important to show that these patients could tolerate, in the setting of an active infection, infusion of NK cells. So there was no cytokine storm or anything like that. All of these patients were discharged from the hospital. And then we have an ongoing trial in relapse refractory AML and MDS, and that study is expected to read out in the last quarter of this year, first quarter of next year. So let me talk a little bit about the engineering technologies that we have. The first is the SNAP CAR platform. This is a universal CAR approach. So this is a novel proprietary technology in which we have a universal CAR, and we can express that in a number of different cells, so both T cells and NK cells. And the idea is to use this in combination and to derive the specificity through tagged tumor-specific antibodies. So one can imagine you can have one bank of a SNAP CAR immune effector cell that you can then give to any patient, and the way you're getting your tumor specificity is through the tagged anti-tumor antibodies. And we also allows us to target multiple antigens and hopefully avoid some of the antigen escape that we see. This is data from Dr. Jason Lohmuller's lab at the University of Pittsburgh, just proof of concept data showing you that indeed with autologous SNAP CAR Ts, you can see specific lysis of targets. So on the upper left, you can see specific lysis using cetuximab with an EGFR target, and then also Raji with the rituximab. And then when you mix these cells, you also, again, can see continued specific specific lysis. In an in vivo model shown on the right with the adapter only, clearly there's no tumor control. This was a NALM6 HER2 positive cell line. So with the adapter only, you do not see any control of tumor. With the SNAP CAR only, you do not see any control of the tumor. But if you do the SNAP CAR plus the adapter, you can see control of the tumor. In fact, equivalent to or maybe a little bit better than just the SNAP CAR, the HER2 CAR T. So this was very exciting for us, and that's the data that we're moving forward with. And we are now we still have active work going on with the University of Pittsburgh, but we've also initiated work with SNAP CAR in the allogeneic NK cell setting. So we are very open to partnership around this technology, and if anyone has any questions or would like to talk about that, just let us know. And finally, the last thing I'd like to talk about is the GEAR CD38 NK cell platform. GEAR stands for Gene-Edited Antibody Resistance. So this is, again, a platform technology, and the idea here is to modify the NK cell by knocking out endogenous CD38, which a lot of people have done. However, NK cells really don't function as well if they don't have a functional CD38. So what happens here is you knock back in an edited CD38 that is resistant to the antibody, however, does provide normal functioning. So the advantages are you can use this in combination. Here, I'm just going to skip to this next one. This shows this much better. So the current pathway here, right, for multiple myeloma or any CD38 positive indication, you can infuse the anti-CD38 antibody. However, what you can see here is that, well, here you have the CD38 expression, the antibody, but this is going to not only cause lysis of your tumor cell, but lysis of an NK cell, which is very effective and helpful in identifying tumor cells. So that's not a great thing. However, when you can do the GEAR editing, now what you have is an edited CD38, which protects that NK cell from daratumumab or antibody-mediated lysis. So this prevents some of the fratricide that we see. So there's a lot of preclinical work going on with Dr. Evren Alici and his lab at the Karolinska Institute, which we're hoping to now bring over into the labs that we have here in the States. So this is just showing you an overview of all of the data that's going on, showing the function of these NK cells, even with the editing that's going on. We have an extensive experience with the management team. I don't have a lot of time. I have 6 seconds left here, so you guys can see this all after, but we've all got a lot of experience here. And in summary, we are hoping to have that top-line data from our phase 1 trials in 1Q24. We're moving forward with the multiple preclinical stage assets that I've shown you, and then we have both NK and macrophage editing that we're working on. Thank you for allowing me to introduce Coeptis to you, and I'm happy to answer questions outside.